This is a stretch of DNA showing relevant regions of gene expression in the Lac Operon in E. coli, a prokaryotic cell. The Lac Operon is an inducible operon, meaning it needs an inducer such as lactose to inactivate the repressor and begin transcription. The Lac-I gene is upstream of the Lac Operon and codes for the repressor which binds the operator on the Lac Operon. It is not part of the Lac Operon itself. The promoter region contains the -10 and -35 box, and on it is the operator, which is the recognition site for sigma and RNA polymerase to bind and begin transcription. The next section is the Lac-Z gene, which codes for beta-galactosidase, which breaks down lactose into glucose and galactose via hydrolysis. Next is Lac-Y, which codes for the galactosidase permease, a membrane protein that brings lactose into the cell. Once this happens, beta-galactosidase breaks down into its monomer subunits. Lac-A codes for the transacetylase protein, whose function is unknown. The repressor is bound to the operator, the normal setting of the Lac Operon, turning the operon off and where glucose is at normal levels. Here, RNA polymerase is guided by sigma to the promoter site as it attempts to transcribe the gene. However, it cannot since the repressor is bound to the operator and thus falls off.
In a different scenario, the glucose levels have decreased because the bacteria's environment has drastically changed, forcing the bacteria to look for a new source of sugar, lactose. Because of the low glucose levels, adenocyclase levels increase, creating high concentrations of CAMP. The low concentration of lactose binds to the repressor, causing the repressor to undergo conformational change and fall off of the operon. Now, RNA polymerase and sigma are able to bind to the promoter. After the initiation sequence is made, sigma falls off, so the cap and CAMP simultaneously bind to form a complex, which then binds to the cap binding site on the DNA, and then onto the RNA polymerase in an energy-requiring process. Because of the already high levels of CAMP, it is able to bind to cap to form the complex. The binding of the CAMP cap complex and RNA polymerase allows transcription to be initiated efficiently. Now the RNA polymerase is able to transcribe the genes in order from Lac-Z through Lac-A. As it makes the mRNA, transcription occurs simultaneously, creating multiple products of the mRNA code because E. coli is a prokaryote which has no nucleus, meaning no mRNA processing is needed.
Looking over to the cell membrane, lactose moves closer to across the cell membrane via the galactosidase permease protein. Once the lactose is inside the cell, it is slightly altered to become allolactose. The allolactose then binds to the beta-galactosidase where it undergoes hydrolysis and breaks into its monomer subunits of galactose and glucose. Glucose as a monomer is quickly consumed as an energy source.
During this process, concentrations of lactose decrease while glucose levels increase. Once glucose levels are at a high enough concentration, the lactose bound to the repressor falls off as the repressor undergoes conformational change again back to its original form and binds onto the operator, turning the operon off. Now that we've seen normal function, let's look at a few mutations. The Lac-I gene codes for the repressor. If Lac-I were mutated, the repressor would be non-functional so that the Lac Operon would always be on and constantly transcribing mRNA. Lac-Z codes for beta-galactosidase. If this gene were mutated, non-functional beta-galactosidase would form, preventing lactose to be broken down into glucose and galactose, meaning there would be no energy source of glucose for the cell. Lac-Y codes for the galactosidase permease protein, which sits in the cell membrane and allows lactose to cross in. If this gene were mutated, then lactose wouldn't be able to enter the cell. However, the system is leaky, and so some lactose might be able to enter but not a sufficient amount. Lac-O codes for the operator. If this gene were mutated, the operator might be missing, mutated, or made incorrectly. This means the repressor cannot bind, and thus the Lac Operon is always on, meaning RNA polymerase will continue to travel down the operon, transcribing the mRNA.